Okay, question 8. There are 3 different types of hemoglobin in the blood of an adult human. Each hemoglobin molecule is composed of 4 polypeptide chains. The table shows information about these types of hemoglobin. Calculate the ratio of polypeptide chains present in the blood of an adult human. Okay, so the ratio of polypeptide chains, and it tells us that we've got 4 polypeptide chains, and it tells us here it is alpha, beta, delta, and gamma. So we want to calculate the ratio of these in the blood. So see what they've given us. They've given us HbA1, and they've told us we've got 96% of this in the blood. And we have equal amounts of alpha and beta. And so what you do is just divide by 2. And so alpha will be 48, and beta will be 48. Moving on to the next one, HbA2. We've got 3% in the blood. And so, again, divided by 2, because we've got 2 polypeptide chains in equal amounts, alpha would be 1.5, and delta would be 1.5. HbF, we've got 1%, again, equal amounts, alpha, so we divided by 2 would be 0.5, and gamma would be 0.5. Right, first step done. So because they want the total of the chains, we've got 4 types of chains. So we've got beta, delta, gamma, alpha, alpha, alpha. So we add all these together to give us 50. So now we've got alpha 50, beta 48, delta 1.5, and gamma 0.5. Now, finding the ratio. Well, the first thing that we do is we divide it all by the largest amount present. So here would be the 50. So the alpha chain is the one that occurs the most. And so we have the highest number for that. So we divide it all by 50. And then we get percentages, just times by 100. So 100%. We get 96%. Times it by 100 to get 3. To get 1. And that's the ratios. And that's it. Right, B. The beta, delta, and gamma polypeptide chains have similar amino acid sequences. The table shows the sequence of 9 amino acids in a part of each of these polypeptide chains. The beta, delta, and gamma polypeptide chains are coded for by 3 different genes. Compare and contrast the sequence of bases in the DNA coding for each of these parts of the 3 polypeptide chains. Right. Essentially, what they're asking for is the similarities and differences. So, we have to compare. Similarities. So here is just state what you see. But the important thing to know is they say the sequence of bases, and here we have the sequence of amino acids. We know that the bases go to amino acids. And so 1 amino acid is 3 bases. So you have a triplet codon. And so here, the first similarity is, well, they all have 9 amino acids. So that means they all have 9 codons, or actually specific triplet codons. And if you wanted to go the step further, we know that because it's 3 bases and we've got 9 codons times it by 3, that means there's 27 bases that make up 9 triplet codons. So hence we have the 9 amino acids. That's first similarity. Second similarity is if we look at the actual sequence, we've got phenylalanine. That's all. Same, and same. So actually, if we're looking at it, then the bases, the 3 first bases, so that's coding for amino acid 1, it's the same bases, and that's also true for amino acids 3, 4, 5, 6, 7, 8, and 9. Oh, no, sorry. Not 3. 4. Because here, it's all the same. So it's only this that is different. So you will say that there are the 3 codes, 3 codons, or triplet codons for amino acid 1 and amino acid 4, 5, 6, 7, 8, and 9 are all the same. And because it's 4 marks, it's usually 2 similarities and 2 differences. Right. But if you wanted to go a step further and say, well, actually, if we look at the beta chain and the gamma chain, they've got the same amino acids for number 2. And if you can have a further look and say, well, delta and gamma, you've got the same amino acids here. So if you wanted to say that, then you would say beta and delta amino acid number 2 is the same, and you would say delta and gamma amino acid number 3 is the same. And so that would mean they would have the same triplet codons to code for the amino acid. So the same sequence of bases. Right, differences. I'll just go over this again. It is this box. Because we've said that there's differences here, and so you would say that the beta and the gamma for amino acids sequence for amino acid number 2 is different. So for beta, it's a different triplet codon to get this amino acids, and for delta, it's a different triplet codon to get this amino acid, and that's the same for amino acid number 3 for beta and delta. And you can say for, let's see, delta and gamma for amino acid number 2. Yeah, you can say any one of those because it shows you a difference. And for the final mark, well, we know that the code is degenerate. And so actually more than one type of triplet codon will code for the same amino acids. So actually, for phenylalanine, you can have UUU or UUC. So this doesn't tell you that bases of DNA, it will just tell you the amino acids. And so actually, they might not have the same bases, but they'll have the same amino acids. And so that's a difference, which is right there. So the bases of the code for the same amino acid might be different. C. Hereditary persistence of fetal hemoglobin is a condition that results from mutations in the genes coding for beta and delta polypeptide chains. People with this condition produce HbF only. The table shows the percentage saturation with oxygen at different partial pressures for hemoglobin F and hemoglobin A. What is meant by the term gene mutation? So that's just something you're going to have learnt. And gene mutation is a change in the base sequence or the quantity of DNA. But usually this is what you would have learnt, and that's an acceptable answer. Right, moving on. With reference to the table, explain why people with HPFH are usually unaware that they have this condition. Well, let's just see what they've given us on the table. So this is with the condition, and this is people who are normal. So let's compare the results. So it seems similar. If not, HbF actually has higher saturation of oxygen. So actually, the capacity for oxygen to be held in the hemoglobin is actually the same, if not higher. So there's no difference in the oxygen carrying capacity of the hemoglobins in HbF. That's 1 mark. So the capacity to carry oxygen is very similar. And so, because this is explained, so what's the significance of this? Well, actually, if it's the same capacity to carry oxygen, gas exchange is the same. Because you'll still have normal processes going on. So actually, you won't have any symptoms, like symptoms usually associated with abnormal oxygen saturation. Normally, if it's low, then you'll have breathlessness, tiredness, fatigue. But actually, if your oxygen is higher than normal oxygen by just a bit, so it's actually normal, then you won't have any symptoms because your body's functioning fine, gas exchange is fine. And so that's the reason. Still have a normal process going on. And that's it.